Hello, everyone, and welcome. My name is Justina Verbover. I'm assistant professor at Cedars-Sinai Medical Center at Los Angeles, and I'm here on behalf of Education and Training Committee under the Human Protein Organization. Together, I'm hosting this webinar with my colleague, Dr. Yanshen Lu, who is assistant professor at the Yale University. At the beginning, he is going to tell you about our committee.
Great, thank you, Justina. So just a correction that I was promoted 2 years ago to be associate. So, but we know each other a long time ago. So we are proud to continue this ETC webinar, which is about the use of proteomics in clinical investigation. So ETC is a committee that was dedicated to assemble programs which include workshops and courses and tutorials that contributes to the professional growth and the scientific awareness of HUPO members. So we are very proud to have a series of speakers in this area of clinical proteomics to share their research and their experience and also some fundamentals about clinical proteomics. So today, we're happy to host Michael Roehrl from Harvard University. So just a little bit introduction about these videos. We actually had, the purpose is not only to have a Zoom discussion, but also to post these videos as public resources for the HUPO community. You can already view the previous series available on YouTube and also in HUPO website. In addition, our ETC committee also will aim to actually summarize these series respectively once we have all the speakers delivered their talk and discussion. So with this, I want to hand the microphone to Justina again, who can introduce the speaker today.
Thank you, Yanshen. So we are truly delighted to host today a webinar with Dr. Michael Roehrl, who is Chief of Department of Pathology at the Beth Israel Decon Medical Center under the Harvard University. And he is a practicing physician scientist, which is amazing. And his clinical and research work is centered at the gastrointestinal pathology. And we are really, truly delighted to have him because his laboratory is using proteomics and proteogenomics to explore cancer biology and autoimmunity. And his team is well known in developing innovative proteome-based biomarkers that aim at creating new generation of the diagnostic and theranostic tools to directly benefit patients. And therefore, we invited him. And as you can see, the title of his talk is Proteomics-Based Precision Medicine for the benefit of the patient. So before that, just a few housekeeping notes. So first of all, please use a Q&A window that you can put your questions, and those questions will be addressed at the end of the talk. Also, we are keeping the microphones off, but just in case, also please mute yourself. And as Yanshen mentioned, the session is recorded, so then you can go and watch it one more time. And without any further ado, please welcome Dr. Michael Roehrl.
Thank you so much, Justina and Yasheng, for inviting me. It's a honor to be part of this and to speak to you all. I'm looking forward to a great discussion at the end. Let me just double check. You can hear me and you can see the first slide.
Everything is great. Yeah.
Terrific. Thank you. So as you heard, what I'm going to try to do in the next 40 minutes or so, 45 minutes or so, is to tell you a little bit about how we view the use of proteomics in the setting of both diagnosing patients prognosticating them, whether a disease is aggressive or less aggressive, and also to identify new markers of therapeutic intervention. I think first of all, I want to congratulate HUPO and also HPP, of which I'm part. I'm actually chair of the HPP pathology pillar, for spearheading this. Bringing proteomics into the clinical realm is very important, very timely. It reminds me of the early days of cancer genomics or genomics overall about 20 years ago, and the rapid advances that have happened in medicine because of this. And I think we have similarly and maybe even more revolutionary discoveries and applications to help patients ahead of us by using proteomics in a clinical setting. So what I want to show you is a motivation, things that I see in my own clinical work. So I'm a pathologist who diagnoses gastrointestinal diseases, and many of them are unfortunately cancers. And what you actually see here are two microscopic images of colon cancer, pretty much middle of the road standard colon cancers. And on the left-hand side is a patient, on the right-hand side is a different patient. The important part here is that under the microscope, these two diseases look, at least to pathologists, relatively similar. But importantly, also, these two patients look based on clinical, what we call staging, meaning the size and the depth of the invasion of the tumor, but also the first, if you will, molecular look at DNA level, identical. So they're both what we call microsatellite stable. They are wild type for common genes that are mutated in colon cancer, such as KRAS, NRAS, BRAF. So they are similar. These two people are similar in age. They have the same disease. They have a T3 colorectal cancer that was resected surgically. But what's interesting is that the patient on the left-hand side was essentially cured by surgery, whereas the patient on the right-hand side, after surgery, which went well, within a few months only developed liver metastases and then died from metastatic disease within a year. So what that highlights is that cancers, even to the best of our current diagnostic technology, that includes all sorts of microscopy, immunohistochemistry, genome sequencing, etc., might look very much the same, yet their behavior, their natural progression is vastly different. And so this is, of course, an opportunity for proteomics or new diagnostic modalities like proteomics to dig deeper and understand the behavior and the natural evolution, but also the therapeutic vulnerabilities of disease at a much deeper level. So most patients, and this is an example, again, you see a photograph here of a colon that's been opened along its long axis. And in the middle, you see this ulcer and this kind of narrowing, and that's an invasive adenocarcinoma. And the arrows basically point to what if you took a piece of tissue from these two sites, from the normal appearing side on the left and from the cancer in the middle, and put them under the microscope, and you would see normal colon cancer. So we're pretty good at making diagnoses. So we can tell the patient what the patient has using technology basically from the 19th century. But what we are trying to get at, and I think, again, proteomics and the integrated multi-omics that we're all talking about, will answer questions that cannot be otherwise addressed. How will the disease behave? Is it going to be less aggressive or more aggressive, like our two patients, which is talked about? And most importantly, from a medical perspective, how will the disease respond to therapeutic intervention? So whether it's surgery, radiation, targeted therapies, et cetera, et cetera. These two latter questions, how will the disease behave, how will it respond, are really the holy grail of oncology. But you can apply them to many other diseases: cardiovascular disease, neurodegeneration, infectious disease, autoimmunity. Very similar thought processes occur, and very similar questions remain unsolved in today's medicine. And again, proteomics, for many of the reasons you guys know about, but also we'll talk about in the talk, has a unique angle at understanding human disease, and will provide answers to these questions that other modalities like genomics or transcriptomics alone cannot do. So this is sort of summarized here, right? From a medical diagnostic perspective, we sort of follow the paradigm of molecular biology, from DNA to RNA to proteins. But most of current applications, at least the ones that are routinely used in hospitals today around the globe, are focusing on the genomics and on the transcriptomics primarily, much less at the level of proteins, certainly much less at the quantitative level of proteomics, large-scale quantitative and proteomics, and also post-translational modifications like phosphorylation, glycosylation, et cetera, are much, much less utilized today than they could be. But that's really what I'm trying to advocate here in the talk, that we need to rethink how integrate proteomics in diagnostics. I'm not going to talk much more about this, just to say that in our Department of Pathology at Beth Israel Deaconess Medical Center in Boston, we have launched what we call a precision diagnostics initiative, of which a multi-omic approach that includes proteomics, metabolomics, the microbiome, is an integral part of the overall way how we are trying to assess human disease, in addition to this more standard next-generation sequencing and some of the other things that we already talked about. So why is this so important? And I don't need to convince you in the proteomics field, but certainly it's an eye-opener to many people who come more from the nucleic acid domains, that actually the correlation between messenger RNA and the actual protein that is being made is poor at best. So this is a study that's quite old, but it shows very nicely that both the steady state and then even more importantly, the variation of protein expression and mRNA expression are poorly correlated, and in some instances even anti-correlated. And that's of course because messenger RNA stability, degradation, production, etc., is regulated differently from the actual translated protein product, and the stability of the protein, the transportation of the protein, the post-translation modifications that the protein may undergo, etc. So it's not surprising, but it's an important fact. And we all know that extrapolating purely based on mRNA abundance to make good biological predictions can be tricky, because the standard assumption, a lot of mRNA means a lot of protein, means a lot of activity of that protein is probably false in many instances. So a good reason for studying proteins directly. And the second one is that a lot of thought experiments could be made, and this is one of them that I like a lot, where you think about two identical twins. If you were to sequence them, the genomes are identical, yet the two individuals are obviously different. They appear differently phenotypically, and one such good example is that the fingerprints of identical twins are different from each other. So one way to distinguish them is really at the phenotypic, if you will, proteomic protein level, and not necessarily at the genome level. That leads us back to the original patients that I talked about that look very similar genomically, right? Those two colon cancers, they are wild type for many of the genes, have very few mutations, yet they behave very differently. And so by extrapolation, this thought experiment, you could have, again, two different cancers or two different diseases that very much look alike from a nucleic acid analytics perspective, but are really different at a protein level and therefore behave differently, have different drug targets, and just from a medical perspective need to be treated differently. So all of that is, I think, very strong evidence for why proteomics is going to be such a game changer when we apply it to medicine. So I don't want to spend too much time on this. You guys are all experts on this, but I do want to say, however, that I think the reason for why there is so much interest in bringing proteomics now much closer to patients than, let's say, even 3 or 4 years ago is because the technology has become much more robust, more sensitive, more stable. The data and analytics and data processes have become more harmonized. And so all of this now bodes well for bringing proteomics as a very sophisticated technology closer and closer to patients, really from a realm of basic discovery, basic science, where it doesn't really matter whether the instrument's down for 3 months and you can repeat the experiment, to a situation where you need rapid turnaround time. You need to harmonize the results between different hospitals, different sites, different laboratories, and you need to be able to run experiments multiple times and compare quantitative changes, and those need to be biologically meaningful. So I think the time is right now to actually think about that in all earnest, to bring proteomics to patients. Something perhaps 5 years ago would have been a little more difficult. So I want to show you a few examples from our laboratory, how we are thinking about moving proteomics more into the clinic. But also, I think important to emphasize that proteomics by itself as a diagnostic tool in medicine will not replace and shouldn't replace the other technologies that are quite powerful, such as RNA or DNA-based analytics. And hence the concept that I talked a little bit earlier about of truly multi-omic characterization and investigation of human disease. I think the common theme across all of this is that these technologies are very quantitative. And if you think about medicine, even today, many of the things we do are very qualitative. We say that we moderate, or strong, for example. Most clinical classifications are very semi-quantitative. The real, I think, advance that multi-omics will bring to patient care is a much more quantitative, number-driven, and therefore computable way how to look at human disease. Now, this is a particular study we did on colorectal cancer. If you remember back those patients that I showed you as an introductory vignette. Why is that important? It's a common disease that has both a primary tumor, here sort of in the illustration, the upper left-hand side, you see the colon as a pictogram, and then the liver. And many of these colon cancers metastasize to the liver, to the lungs, and other sites, and that's really what kills the patient. So this was a large-scale colorectal cancer study that we published recently, where we used proteomics both in the primary tumor and the metastases, and tried to identify proteomic subtypes that were not visible based on genomics alone. And we identified a couple of different signatures, and I'll walk you through that in a moment here. So what's, however, really interesting is that if you compare normal, that would be normal colon lining, primary tumors, so those are cancers that sit in the colon, and the metastases in the liver. If you compare them at the RNA level, interestingly, you can't distinguish primary from metastases. You see in this UMAP plot on the upper left-hand side that those cancer domains or clouds really are indistinguishable. Yes, normal is different, that's not surprising, but the cancers are not distinguishable. But if you start doing this at the protein level, you actually start to see that metastases and primary tumors tend to be different. And I don't want to go too much into the detail of if you will, the heat map on the right-hand side. This summarizes what we called our discovery cohort at the time, really showing the three subtypes in primary cancers on the left-hand side, and in the right three columns. These are proteomic subtypes of liver metastases. And we learned a lot from it. First of all, that primary tumors in the colon and their matched metastases are not necessarily proteomically identical. Actually, there's quite a few differences. However, at genome level, the number of mutations between the primary tumor and the metastases is essentially the same. So there's no significant acquisition of further genomic drivers. It appears more that the proteomic rewiring or the signaling rewiring differentiates primary cancers from their matched colon liver metastases. So these are some follow-ups with specific protein markers that characterize these different signatures, such as a hypoxic signature, a cancer stemness signature, and so forth. And you can also see that for some of them, for example, here in the stemness signature, if you focus on that, or some of the oncogenic functional driving signatures, primaries and metastases are significantly different. So it's not the underlying genomic architecture of these cells that differ, but it's really the way how they signal or how they function at a functional level. What's also fascinating is that not every primary subtype 1, if you will, becomes a metastatic subtype 1. There is dynamics and fluctuation built in. So that's very interesting biologically, because it also means or suggests that by just looking at the primary tumor, it's not probably impossible actually to predict what the metastatic phenotype or proteomic type will be for a particular patient. And that is very important, because it's likely that these different subtypes that we identified need to be treated differently because they have different therapeutic vulnerabilities. So that highlights really that from a medical perspective, it's going to be very important to test and analyze the lesions that are the most disease-causing. So metastatic disease, and even among different metastases, sometimes the ones in the liver grow fast, whereas the lung metastasis is stable. So importantly, we as clinicians need to decide which of these lesions to biopsy and analyze to make the most impact therapeutically. And again, the assumption that's currently made clinically is often that you can predict the behavior of metastatic cancer by just looking at one site. And I think that's clearly wrong based on our proteomic data. When we dig a little deeper into the proteomic subtypes of these liver metastases, these subtypes, we actually find that at a protein level, when we do network analysis and gene enrichment or protein expression enrichment analyses, we find both shared protein pathways that are upregulated, that are shown on the left. So these pretty much every colon metastasis in the liver will have these activated, whereas then there are also some that are specific only to the metastases and differentiate the metastasis from the primary tumors. And if we then drill a little bit deeper, even among these different metastatic subtypes, you see that these three types are characterized, and that's obviously the correlate, if you will, the underlying driver for why at a proteomic level we find these subtypes. There are different pathways, different mechanisms that characterize each of these metastatic subtypes, these types of liver metastases. And just by looking at it, you will see that obviously this would likely imply that there are different therapeutic vulnerabilities available in each of these subtypes. And knowing which subtype the patient has would likely drive therapeutic intervention. So this is an in silico experiment we did, not yet validated in patients, but the idea, if you took these individual subtypes, how many of these would potentially be targetable, and actually the targetable drugs, of course, would differ between these different subtypes. So I want to just return briefly to the two patients I talked to you about, and we've actually created a, if you will, a proof of principle multi-omic cancer assay that includes a proteomic component. And it turns out that the two patients I just talked about at the very beginning, if you remember, they're identical genomically, and otherwise, similar age, similar stage, etc. But when you incorporate a proteomic profiling of the cancer, they actually are different. They're different types. They fall into different subtypes of what we call microsatellite stable colon cancer. And so what this would hopefully now tell us is that, yes, indeed, these patients are different, and would open a door to then say, well, who is the more benign, the sort of the course, the patient who basically was cured from surgery alone, whereas which one is the more aggressive type that perhaps would have benefited from a very aggressive treatment right up front before the liver metastases appeared. I want to switch gears now to a different application to show you again sort of how we could use proteomics in now not a very common disease, but a group of rare diseases for which genomics actually hasn't really been so successful, because either these diseases don't have well-known driver mutations, or they have very few mutations. And what we looked at is actually a collection of 4 different pancreatic tumors. They're listed on the right-hand side. Don't worry about it. They're rare, but they're important because, again, very little has been gained purely from a genomics perspective. And these are different from the typical acinar ductal adenocarcinoma that most of you will be familiar with. These are rarer tumors. And especially one of them I'll talk a little bit more about. That's neuroendocrine tumor of the pancreas. And if you remember Steve Jobs of Apple fame, he died of that tumor. They tend to be relatively slow growing, but what's really difficult is that they do not have many mutations, and none that are really recurrent, and virtually no therapeutic targets at this point. So we did, standard, if you will, proteomics, very quantitative. I think what you will also recognize once you bring proteomics into the clinic is that you often have to work with very suboptimal samples. So for rare tumors, we often don't have them fresh, frozen, highest quality samples. We have to use these older so-called formalin-fixed paraffin embedded samples that exist in large archives in general pathology departments. Because they're rare and they often are accumulated over many, many years, as was the case in our case. And so the extracting the proteome, doing that somewhat quantitatively without introducing too much bias or error is not necessarily an easy task, and that's something one should always be aware of. This is sort of the bigger picture when we look at total proteins and what are perhaps some entity-specific proteins that are primarily expressed in one of these 4 diseases that are listed here. So they do exist. If we just look globally and say, well, we're not a pathologist, we're just using computer science to tell us what is what diagnosis, you actually see that the computer, just the proteome, if you put it in without the diagnosis and let it separate into clusters, actually can diagnose these 4 diseases very well. And what's also really assuring is that in red, the acinar cell carcinomas, ACC, and the green PBLs, pancreatoblastomas, those are childhood cancers. The acinar cell carcinoma, very aggressive tumors of adulthood, but they share actually under the microscope a lot of features with each other, and they tend to cluster closer together here in the middle. And that's quite reassuring, whereas the neuroendocrine tumors on the right-hand side or solid pseudopapillary neoplasm on the upper left-hand side are really very different beasts. So without learning any medicine or any pathology, the algorithm actually picked it out. And so that means to me that the underlying proteomic signature of these diseases is a very strong informant of the actual disease, and recapitulates essentially what pathologists have been learning over 100+ years, how to categorize these diseases in, again, in a totally unbiased way. So what I'm showing you here is a comparison between acinar cell carcinoma and pancreatoblastoma. I already told you they sort of look alike under the microscope, and if you look at the two images on the lower left-hand side, you will probably say, oh yeah, they do look alike. There may be some tiny subtle differences that if you're a pathologist you would pick up, but generally they look alike. But they do have very distinct protein signatures that actually make acinar cell carcinoma of the adult and pancreatoblastoma of childhood two different diseases, albeit related, but there are some very clear distinguishing factors. And we can actually even surmise that pancreatoblastoma itself, which is, again, as I told you, an interesting rare tumor of childhood, may even have two different subtypes. And that may be related to different pathways that I don't have too much time getting into today. But again, things that proteomics can uncover that standard pathology has so far not seen or not been able to discover. Briefly about solid pseudopapillary neoplasm of the pancreas, something we've been studying for the last while in the lab quite a bit. It's a very interesting rare tumor. Very little has been known about the genomics, except that Wnt signaling and mutations in beta-catenin are very common. This is a tumor that's very much more common in women. It's a slow-growing neoplasm, but proteomics actually rediscovers, and that's not very reassuring. If you look on the lower left-hand side, the fact that both beta-catenin and LEF1 are driving this tumor as opposed to control normal or carcinoma of the pancreas and neuroendocrine tumors. So basically, this is validating the biology, the little bit of biology that was known about the tumor. But we've since gone on to do a lot more work around metabolic adaptation, the immune infiltration of the tumor, and even features around autophagy and certain granule formations that occur in these cells, understanding what happens, for example, to the presentation of autoantigenic peptides, why these tumors are so poorly recognized by the immune system. So there's a lot of interesting biology that proteomics has been able to uncover on top of validating essentially what genomics has found, that make this disease that remained a big mystery now much better understood. Now I want to get back to these Steve Jobs neuroendocrine tumors of the pancreas. They're slow growing. We call them well differentiated. They have very few mitoses, very little, but they are insidious. They grow slowly and then they metastasize to the liver, and then they're essentially not curable. And again, do not have recurrent mutations or clearly targetable molecular vulnerabilities for which drugs are currently available. So similar to the story that I told in colon cancer, there are people who are cured, and then there are people who are not cured, like Steve Jobs was. And currently, frankly, it's totally impossible to predict. What happens is patients have surgery and then are followed by radiographic imaging, CAT scans or MRIs, and essentially you wait for the tumor to regrow or to metastasize, to declare itself basically before you do anything. And that's, of course, very stressful for patients, and it's also very unsatisfactory in terms of medical care. So we try to figure out, can we use proteomics to actually learn more about these neuroendocrine tumors? And again, I think I mentioned that already, they have very few mutations, so they're mutationally rather silent. And so traditional genomics has not been so successful. So we did a larger neuroendocrine tumor proteomics study. Again, it's a multi-omics study. We also looked at genome sequencing, copy number alterations, but also did proteomics, and actually found 4 different subtypes, 2 of which are clinically unfavorable and 2 are favorable. We also identified things like an hypoxia signature, an immune signature, how some of these tumors evade immune recognition, and also identified possible therapeutic targets for each of these proteomic subtypes of pancreatic neuroendocrine tumor. This is the bigger picture here. We see 2 groups, P1, P4 on the left-hand side. They are a little bit different from each other, but somewhat related, and then P2 and P3 on the right-hand side, and correlating them with mutations such as ATRX or DAX. So there's no real difference between the two. So it's not that the genomics is driving it in any way. The gender distribution, the age distribution is also similar. So this is something that's intrinsic to the tumors and independent of other confounding variables, if you will. But when you look now and group at the subtypes, the neuroendocrine tumor subtypes 1 and 4 together, and 2 and 3, you see that 1 and 4 are really of indolent type. So if you look at these Kaplan-Meier plots here, the probability of recurrence-free survival is almost 100% for the 1-4 subtype, whereas it drops significantly for the 2-3 subtype. And we have fairly long follow-up, as you can see, of well over 60 months for these tumors. And that's very important because, as I told you, they are slow growing. And so a clinical follow-up time is really crucial to make this difference or show this difference very, very, very clearly. And then you, of course, can ask what actually biologically differentiates the better outcome subtype on the left from the more aggressive subtype on the right. And you see the volcano plot, there's a number of protein expressions. And if you put them into pathways, so there are a number of things that make a tumor more aggressive in its behavior, such as hypoxia and a number of other things that I really don't have time to get into now, but this is also published work. And that are not visible under the microscope. These tumors do look the same, so it's hard to tell the difference unless you do proteomic analysis of these tumors. And that's sort of shown here, where we show that 1 and 4 have a much lower hypoxia score, so the good outcome from the bad outcome 2, 3, for example. So there are features that make these differentiated. There's a number of other biological things like enzyme expression in glycolysis, TCA cycle, etc., that differentiate metabolically the better outcome, or if you will, indolent, what we call indolent neuroendocrine tumors from the more aggressive ones that progress. And similarly, there's an immune signature. So it turns out that the more aggressive subtype is considered more immune hot, which is quite interesting, despite the fact that it actually isn't recognized by the immune system. So there are things that obviously this tumor can do to reduce the activation of a T cell immunity, for example, despite the fact that it actually does present antigens quite effectively. So what we also then did is we tried to figure out, can we use proteomics to identify possible drug targets in these currently essentially untreatable neuroendocrine tumors. And yes, of course, you can try to do that by comparing, again, the benign pancreas versus the different tumors, but also asking what are some of the druggable targets or pathways that specifically characterize the more aggressive subtype variants. And that's sort of shown here, where you see potential drug targets, both the proteins and the types of drugs shown here in this matrix for the better outcome type 1-4 in the top and the 2-3 more aggressive in the bottom. And while this remains to be clinically validated, I think it is at least a proof of principle of how you could use proteomic testing for a group of patients or even individual patients and trying to map their proteomic profile to potential drug targets and druggable vulnerabilities. Another finding that came out of this particular study was very interesting. The common wisdom has been in the field of neuroendocrine tumors that counting mitoses, basically saying if the tumor is more mitotically active, it'll do worse. Or conversely, if it's mitotically more silent, it'll do better. That at least for the so-called well-differentiated tumors, these are the ones we're looking at here, that tend to have relatively low mitotic activity to begin with, that actually this grading of G1, G2 may not matter as much as the actual proteomic subtype for predicting outcome. And that's shown in the upper left-hand side. If you look at subtype 1 and subtype 2, the distribution, if you will, of G1 and G2, grade 1 and grade 2 tumors is almost equal. So there are both G1s and G2 in subtype 1, and there are G1s and G2 in subtype 2. But if you remember, the subtype 1 was the one that didn't have any recurrences, whereas pretty much all the recurrences in progressive disease was in the subtype 2. So it's not just the traditional assumption that mitotic count matters. There may be more to it. And I think this is, again, an example where proteomics can add significantly to the current state of best knowledge that we use in tumor diagnostics. As another example, I would like to show something a little more functional, how proteomics could be used to do that. And this is work from a postdoc who is a thoracic surgeon and therefore very interested in lung cancer. And for those of you working on lung cancer therapy often starts with tyrosine kinase inhibitors because many of these tumors are sensitive to them, although most of them ultimately develop resistance. And resistance to these drugs, to these small molecules, is a major clinical obstacle. And what we were trying to do is understand, can we model the resistance and use proteomics to figure out why certain tumors become resistant and maybe even use that to overcome resistance. So we actually looked at syngeneic cell lines that were inherently sensitive, but then were in vitro made resistant over 2 to 3 orders of magnitude of this osimertinib, which is an advanced category tyrosine kinase inhibitor, to figure out what makes these cells either sensitive or resistant. And we did whole exome sequencing, of course, of these cell lines as well to figure out whether underlying genomic alterations would be drivers of resistance or not. And we can actually show that essentially there aren't any additional mutations if you make them, at least these cell lines, if you make them in vitro resistant by growing them in increasingly higher concentrations of the drug. So it's not the genomics that would explain it. There must be something else. So we looked at both total and phosphoproteomics with and without treating these cells with tyrosine kinase inhibitor, try to figure out some of these differences. And that's shown here. So these are just two of these patient-derived cell lines that I'm showing you. And you can see that with increasing concentration of the drug, the sensitive cell line starts to die, whereas the resistant cell line maintains viability, or at least over 2, sometimes 3 orders of magnitude concentration increase over the sensitive cell line. And so then we ask questions, how are they different, both at global and at phosphoproteomic level, both in the presence or in the absence of TKI treatment? And long story short, we were actually able to identify pathways that appear to be upregulated in the resistant lines that may render them resistant. And if we now target these alternative drugs together with the TKI inhibitor, we can actually create a synergistic effect by targeting 2 pathways that are unrelated in this particular case. And that's shown at the bottom, where you can start killing these cells, even though they're highly resistant, by targeting another, if you will, synergistic pathway. And the identification of that synergistic pathway came from a quantitative proteomics study. So you could again envision, this is interesting biology, but think of this as a concept for patient care, where you could take a biopsy from a patient when the patient developed resistance, and then identify alternative drug targets ex vivo that you can then bring into the patient to treat. So this is sort of, again, a proof of principle example of how proteomics could be used, similar to the way, for example, in antibiotic therapy, people do antibiotic resistance measurements and then give the patient the right combination of antibiotics because they already tested the patient's bugs ex vivo in microbiology before going back into the patient. This is applying the same idea to oncology. I don't have too much time to talk about this, but this is a little bit different here, methodology. So one of the challenges we face in clinical proteomics is that oftentimes our cohorts are small or seem to be biased by a lot of confounding variables like patient age distribution, gender distribution. So if you look at a disease with high risk and low risk and want to figure out which markers tell us whether the disease is aggressive or less aggressive, if you actually use real clinical groups, they have inherent biases and inherent distractors built in. So, one group might have many more women than the other one. And so that by itself could be a driver. And all you're identifying is essentially the biases that you have. And you can't really avoid them because you have to use, in clinical medicine, you have to use the cohorts of patients you have. So there are approaches to try to do this. One of them is called propensity score matching. That's been used in clinical trials sometimes, but not so much yet in the field of biomarker discovery. And the way it works is that you actually take from your entire cohort a subset of patients and you match them for as many of the confounding variables as you can. And so you end up, in a way, a little paradoxically, with a smaller group, but the group itself is much more homogeneous, and you're removing a lot of these biases. And so we've published on that. This is sort of how to show you. You start at the top with a large group here, in this case, colon cancer, but they're really not matching well. You see, the propensity scores for the good and the bad prognosis group is very different. So you're going to find a lot of confounding variables if you do multivariate analysis in addition to the proteomic biomarker you want. If you look at a much smaller cohort at the bottom, so we extracted that, but matched them, now they're much more matched. And so even though you're using fewer patients, that actually means the cost of the experiments goes down because you need to analyze fewer. But the differences that you then have are going to be much more pronounced and not as much confounded by demographics or other kinds of comorbidities and so forth that might bias you otherwise. And we'll show that here, where before the matching and after the matching, you really discover basically things like, TNM staging, yeah, it doesn't matter how big the tumor is, or whether lymphatic invasion or perineural invasion in the tumor matter. Yes, they do, but you already knew that. So you want to remove that. So if you look at the column to the right, after propensity score matching, all of these known factors go away. But at the same time, the number of possible protein biomarkers that seem to correlate with good or bad prognosis also is reduced. As you can see, a couple of these markers were actually spurious. And once you do propensity score matching, STAT1, for example, signaling, which is important for outcome in colon cancer, remains as the most powerful biomarker at a protein level, whereas the others really are removed because they more likely correlate with other features, but not the prognosis itself. So in the last few minutes of the talk, I want to summarize and give you maybe a little bit a bigger picture of how we think about protein-based diagnostics, proteomics in a clinical sphere. I've talked a little bit about things like genomics, for example, and clinical trials perhaps a little bit too. They will remain, but adding proteomics, adding digital pathology, adding multi-omic approaches will really change the way how we diagnose and analyze human disease in the next few years to come. It's also important, I think, in clinical medicine to always strive to shorten the time it takes from the research bench, from discovery to the patient benefit. I think proteomics has been slow in coming. Sometimes perhaps because instrumentation is tricky, not stable, and so forth. But once the tide turns, once the inflection point is there, and all of these technical barriers are removed, I think the adoption of new technology like proteomics will be very, very fast in clinical medicine, and it's like with genomics. Nobody would say, oh, let's go back 20 years and we don't need it. No, it's become such a mainstay of modern medicine, and I think the same will happen to proteomics in the next few years to come. I wanted to say a few things about that. Our own organization is building together Dana-Farber, a new cancer hospital. And so for us, of course, we spend a lot of time thinking about what is the future of cancer diagnostics, what are some of the new tools and technologies to reimagine cancer medicine of the next years and decades to come. And integrated proteomics, multi-omics, also very quantitative measurements of post-translational modification, signaling events, etc., both at a bulk and also at the single cell level, I think will be extremely important for moving cancer care forward, for running new clinical trials, for developing new drugs, and so forth. So we put this whole umbrella, if you will, of proteomics and multi-omics under this label we call precision pathology. Why precision? Because the measurements are intrinsically quantitative. Ultimately, how many moles of a particular proteins are there on an individual cell, or how many copies of a particular protein, or what's the percentage of a protein that's in the on state versus the off state, how much of a protein is correctly multimerizing or dimerizing with a partner, how much of it's in the right place, like a transcription factor in the nucleus versus the cytoplasm. These things are, amenable and measurable by proteomics. And as we do that more and more using this quantitative data, that's really what we think of as precision pathology. And we think the more precision you have, the more precisely quantitatively you measure something, and ideally even repeatedly during the course of disease with and without treatment or at recurrence, the less a patient has to fear a disease, because the more you understand it, the less you have to fear it. The worst in cancer is if you say, well, we think it's malignant, we think it's bad, but we really don't know what it is. So I think these tools like proteomics have the capacity to bring a lot more light into the dark of patients and understanding their diseases. So these are some of the papers I talked a little bit about, the pancreatoblastoma paper, the colon paper, the neuroendocrine paper, and so forth. I should also say that obviously there's a big effort going on in many countries in the United States and worldwide to bring proteomics to patients, to develop the tools, to develop the software, to make these things robust. And that's very important. And I think HUPO and HPP are doing phenomenal work to advance this. I would like to invite you all, if you're interested in the concept of applying proteomics to human medicine, to reach out and ask about our pathology pillar. We will have a workshop at the HUPO meeting in Toronto on Sunday, and we will also say a few things during the HPP day. And again, this is an invitation for the community to get involved, because if we get involved as a community and drive the technology and bring it to patients, it will happen. If we don't do it, it will not happen. So a very important aspect of how do you bring a great technology, a great idea to the benefit of patients. I have a lot of people to thank. A lot of the work I talked about today was Atsushi's work. Atsushi Tanaka, Makiko did a lot of work, Masaki on the propensity score matching, and Yusuke. Kei Namba is the thoracic surgeon who worked on the cell line model. And last but not least, I want to thank all of you for inviting me again to give this talk today. And I'm looking forward to questions. Here is my contact information if you want to reach out. So again, thank you very much.
Thank you so much. It was a very inspiring talk, and I personally learned a lot, and I have a few questions, but we also have a question at the Q&A, but if anyone would like to put their question or answer in person, you can just raise your hand and do so. Yenshen, do you want to take the first question? So
so we could also enable them to talk. If Jay, you want to ask your question, you're welcome to do so directly.
Okay. Okay. Thank you. Can you hear me, Sean?
Yes.
Yes.
All right. Sorry, it's too late in China, but very honored to ask the question. Well, I actually wrote something, made my notes in the chatting area, but I recalled it as the first one is, have we what is this? Have we applied microdissection for this work? That's the first question.
Yeah, that's a great question. The things I showed, no, most of that was done out of bulk. Although I should say, as a pathologist, you have an advantage of which disease you study. So, for example, neuroendocrine tumors, when you take them and grossly look at them already, they're going to be 95+% pure. Similar acinar cell carcinomas, pancreatoblastomas, etc. If you study, for example, pancreatic ductal adenocarcinoma, where as tissue volume, maybe only 5% are actually cancer and 95% is inflammation and fibrosis and all of that. I think your question is right, spot on. We certainly try to pick things where it's a little bit easier. We've done laser capture microdissection also. It's of course much more tedious, and you have to work with less, and it becomes so I think your question is absolutely spot on, and it depends on the disease you're studying. We also have experimented with flow sorting, of course, and tissue disaggregation, and ultimately, of course, the idea to do things more at single cell or just a few cell levels, which again is doable in our own hands. And I didn't talk about it here. The requirements on the specimen quality become higher and higher. The less input you use, if you go down to single cells, it becomes so important, especially if you work on material from actual patients as opposed to cell culture, that your samples are really procured under the most standardized, reproducible conditions. Otherwise, you're going to find lots of variability, but you're never sure whether it's post-analytical or right. And so that is absolutely a good question. But it makes the pathology point of and the medical aspect of the tissue and the disease you study even more important.
I see. The second question, if I may, well, because we have like during the discovery stage, you applied, I see a couple hundred, I guess, cases, and it's a long run for mass specs. And my question is, how do we normalize data between runs or between patients? And how did we apply QCs for those like semi-clinical application?
Yeah, that's a phenomenal question again. And I invite you to look at the papers and, Atsushi and people whose work I show primarily spend a lot of time. It's also, I think, very elaborately described in the papers how this was done to try to make sure that there isn't much of I mean, first of all, you mix and you do things just in an experimental setup, but then of course you try to look at the data and harmonize it. Is there a perfect approach to it? Probably not. I think you have to be aware of it and you have to try to see if there's batch effect and things like that. In the future, I think, a lot of this will be run much in faster gradients and many more samples in a short period of time. So perhaps that'll take care of some of that. I also think that most of what I talked about is still a little bit in discovery and researchy. If this is used routinely on patients, we may not need, the full proteome or the full characterization. We might rather look at certain proteins more quantitatively by using also internal standards, labeled peptides, et cetera, right? Things that have quality control built in. And that's of course borrowed from a lot of what laboratory medicine already does. When you run, a lab assay for a tumor marker today, often, using an antibody-based method or so, the batch effect and sort of the laboratory validation, et cetera, is front and center. I think this will be something that proteomics is doing already, but I think a very, very valid point.
Surely. I have my clinical lab and I try to apply a genomic application and the proteomics, put them both for like a target drug selections. However, one of the struggling part is we categorize the patients into different subtypes, but that's based on a cohort. We have to normalize the whole data and to make a panel or algorithms. But when the new patient comes, that's a single patient. How do we normalize it again? That's usually always my struggling part. Do you have experience on that?
Yeah, that's a really great question. I think this is particularly relevant if you look at patterns, right? If it's a particular pattern that differentiates, let's say, a good outcome from the bad outcome. Of course, if you can try to hone in on certain pathways that actual biology that differentiates the two, then I think you have a better chance because you could try to quantify that pathway by mass spec more quantitatively, right? I think this is an inherent, what you're describing is an inherent problem of using large scale data and then using it more as a pattern recognition, saying, okay, this pattern goes with this and this, pattern meaning a bunch of proteins up or down, right? I think the approach I would take is, to make it an actual clinical test. You probably want not the pattern to be the driver, but you want to use the pattern to discover the pathways and then pick members of the pathway as an actual measurable that you measure in the routine clinical assay.
Okay, thank you. It's a pleasure.
Great.
Thank you so much, Jay.
Thank you, Jay.
I will follow with that question because I think so you answer in some respect my question about this not particular proteins but the pathways, and we know that there is a pathway and there is sometimes hundreds of proteins. Yeah, so you show a really nice slide, and you pick some of those proteins. So because you're also physicians, yes, in your practice, if you have a patient, do you do that? That, for example, you know from your bigger study that that and that protein, it shows some potential. Have you ever done something for your patient that you check if that patient had a particular 5 of those proteins, something like that?
Yeah, so maybe the most specific examples are we have identified, so in solid pseudopapular neoplasm, for example, we have identified certain proteins that we then followed up by immunohistochemistry, and immunohistochemistry is much easier to use in routine pathologic diagnosis. So we've actually developed some markers that single or groups of markers that people now can use, for example, to make the diagnosis or to stratify more aggressive from less aggressive disease, and don't have to run necessarily a full proteomic panel on a patient, but can use a smaller set of a few markers. And those are, some of them are known, but like in SPN, it's actually a known marker, but it's never been used in this disease. And sometimes they are, available markers, but they've not been used at all. Like in colon cancer, we have a few where we can differentiate the outcome of aggressiveness. And that those are really distilled down, if you will, from the total proteomic discovery. And then, using immunohistochemistry, of course, much cheaper, much faster, and you can do it routinely already. So yeah, we have a few of those in colon and some of these solid pseudopapular neoplasm, for example, the pancreas, that we now can use clinically.
Great. I think Giuseppe actually had a following up related question. So Giuseppe, do you want to ask it yourself, or I can read it for you? So basically, Giuseppe's question is about these immunohistochemical parameters. So Mike, so his question is that how many patients do you need to implement this type of scoring, or if these scores are cohort specific or universal for all the patients in the same cancer?
Yeah, that's a great question. Again, I don't know how many patients. I mean, more is always better in principle. So we oftentimes, for colon cancer, for example, we have tissue microarrays. I mean, in addition to what we do for proteomic discovery, where we validate these things, that are several hundred patients, 300, 400 patients. For our neuroendocrine tumors, it's fewer. We work also on sarcomas, and they're just limited. You pretty much try to get whatever you can find. But yeah, those are much larger cohorts, of course, where you try to validate and independently validate. I think the real bottleneck in clinical proteomics remains still good sample sets that are not only the quality of which is good, but also the clinical annotation is robust. Because if you are trying to correlate, again, good bad outcome, etc., if the annotation is not solid or not validated or comes from a homogeneous background, and you use multiple hospital sites and they call things differently or so, that of course introduces a huge bias potentially. So we spend a lot of time trying to figure out, is the clinical annotation correct, not only the stage, but especially when you talk about survival and recurrence-free survival, right? That it's real. And that, I think, remains for all of us, even for those of us who work in hospitals where these materials are actually coming from, remains a major challenge, I would say, especially for rare diseases.
Great. So Mike, I also have some questions. I mean, you actually gave a very nice introduction about lot of applications, different cancer types, cancer cell models, and even data analysis, and your visionary views for the future. Because I think that's very much appreciated, because the goal of the ETC is also to have some fundamental discussion and involve the students and for education. So I want to take a step back to maybe ask you about some technical basics, like in your lab or in your experience, because for CPTech they normally use TMT, and what type of mass spec and data acquisition method are you using, DIA or DDA? Do you have some experience to share with the audience?
Yeah, so that's also a great question. Yeah, and again, it's in the papers, people can look it up. I mean, we are not a lab that does real methods development or develops new methods, right? We use pretty much what So we've done similar. I mean, our initial approach is very similar to what CPTAC is doing with the multiplexing. We're using commercial kits to do it. I think our unique approach is more picking the right disease, asking the right questions on unknown, and sort of the question earlier: do we microdissect? Yes or no? We are not, and actually, I'm thrilled. I have some local collaborators, but I would be thrilled to collaborate more with laboratories that are more on the technical development side, especially as we talk about more single cell proteomics, some difficult PTMs. So we have a fair amount of experience with phospho. I didn't talk about that here with phosphoproteomics, but looking at more unusual post-translational modifications, glycosylation is very tricky, I think, in our hands to do. So while I'm not, perhaps the best person to talk about what's the most cutting-edge technology to develop, I think our strength is that we apply, what I think is still pretty cutting-edge technology, but to the right disease or to the right sample. And also recognizing limitations, like when we do bulk, then we have to go back, or we do cell deconvolution. So for example, we've done some work on using bulk, but then using in silico, trying in silico to deconvolute the cell composition. That's what we're doing in the sort of immune space a little bit, to figure out which proteomes came from the lymphocytes, which ones from the cancer cells, which ones from vascular cells, similar to what people are doing in the mRNA space. And I think we're very transparent. So our papers are very clear about what we use, the methods, software, all of that. But I think our strength and what I was trying to show here is ultimately, I think, it's like with any technology. It's like if you think about sequencing too, there are many ways how to sequence, many platforms, right? Many chemistries. But the real excitement about genomics, came about when people started using it on interesting disease or interesting cancers. And I think that's sort of how we see our group, is to push. And that's why that's why HUPO is so great, I think, as a community, because it brings together both the tech people, like both from the software and the hardware and the chemistry side, but also from the medical side, together to push the field forward.
Yeah, that's great comments. So answer the correct question using the techniques that are available, right? To see how that would change the landscape. So yeah. I find the comment about the size, the cell type specific features and the cell composition is actually very important consideration. If you look at the PCA result or your UMAP virtualization, so how many of these variants are really come from the cell type specific proteome, or is really related to the cancer diversity, right?
Yeah, that's a great question too. And of course, I mean, one advantage we have being in a pathology department is that we can try to at least standardize our samples as much as we can in terms of tumor content, morphology, etc. And that's why also the diseases I showed you, they are a little bit in our favor, because many of them are what we call high cellularity diseases, meaning even in a natural human being, these tumors tend to grow very cellular, so they don't have a lot of stroma, they don't have a lot of necrosis, right? So they don't have the features that of like breast cancer, for example, or standard pancreatic cancer. They are notoriously difficult to study because sometimes the cancer is only a small amount of the tissue, and the rest is all sorts of other things like fibrosis and vessels and inflammation. And then you have huge variability between people, or even if you sample within the same patient, you get huge variability, and you're never quite sure, is this cancer, or is it just because it's just geographic differences that. So what I showed, I think we are fortunate because we can sort of prime ourselves for some success while minimizing something. That doesn't apply to all diseases under all circumstances. And that's why I think some diseases are just so much harder to study than others.
Yeah. My final question is related to the future. Like, what do you do after the proteomic discovery, right? You nicely mentioned that there are certain important proteins which are targets for multiple FDA drugs. And in your osimertinib study, I think it's a very nice design that you could compare the difference and learn the difference and maybe try to predict some drugs, right? So yes. What do you think the next step is once you have all of this proteomic information?
Yeah, I know this is a fantastic question. I think and not just for us, I think for the field, what will make a real difference is once proteomics becomes an established component of clinical trials, because then you can start both to correlate findings with actual outcomes, and you can look at intervention. And the idea is also that you could use, certain markers. I mean, in the osimertinib case, of course, right? Ideally, a trial where, you have an arm that allows you to do an intervention once you detect the resistance, for example, and follow that resistance at proteomic level. So there's, I think, two things we have to overcome in the proteomics field. One is we need to get proteomics much more to become a routine part of ongoing phase 1, maybe also phase 2, but certainly phase 1 clinical trials, which it currently isn't. Genomics is, but proteomics is not usually part of this. And the second is that we have to push for proteomics to become an established companion diagnostic, meaning that you need it to demonstrate the presence or absence of a particular actionable marker. Current standard is still that immunohistochemistry or genomics are the two prime examples of companion diagnostics, right? Pretty much every companion diagnostic out there is either one or the other. It's usually an immunohistochemical assay or some sort of genomic assay, or maybe a FISH probe. But once proteomics becomes an established tool to actually measure, imagine a time it has proteomics and you measure HER2 levels, and maybe even the fact that HER2 is on, or something is on or off, a kinase is on or off, right? Rather than how much of it is there. I think then the switch is turned, that proteomics becomes a really valuable tool that we can't be without anymore.
Great, great. Thank you so much, Mike. So Justina, do you
Thank
have
you.
more questions?
No, thank you so much. I have the similar question, how to shorten the time, yes, from the bed to the lab and to come back to the patient. And I think so, you just said that, I think so it's still in progress, yes. And I think so we have to push it, we as a community, to be more standardized. And I don't, I envision to do the PRM and MRM assays, as this particular tool just go very quickly, like now it is with the genomics. But hopefully, in the future, quick future, we'll have this one. And I hope so that all of our attendants and people who are joining us later are going to agree with me and work on that very hard to include those assays in our clinical portfolio. So I think so with that, yes, Yinshen?
Yeah,
Thank you so much. Thank you.
Thank you, Mike, for this great lecture and a fantastic discussion. Thank you, everyone. So please keep an eye on our HUPO ETC webinars in the future. This webinar is over, and thank you. Have a nice day. Goodbye. Thank you, Mike.
Yeah. Thank you so much.
Bye-bye.
Bye.
Thank you.
Thank you.